All right, hello again. The next AP Biology review video, as you can see, is going to be about the cell cycle. There'll probably be two of these because there's a lot of details and I don't want this to go too long. We'll see how that goes. But this is all leading to something called cell division, right? We want to make new cells. And so the first question we should ask ourselves is why we wouldn't want to do that. And right now is a good time to stop and remember that there are other kinds of organisms out there that are not like ourselves. These would be single-celled organisms, and things are going to work a little bit more differently in them than they would in an organism like us. So over here on the left side of the screen, you have a picture of something called binary fission, which is how a bacteria or some other prokaryote would reproduce. This is where they would make a copy of their DNA and then just split the cell in two to make two cells. This is easy for them because they only have one chromosome. But then we have eukaryotes, and some eukaryotes are single-celled organisms like protists, which include parameciums and amoebas and things like that. They have to go through something called mitosis, where we have to do these steps in order to move around and segregate our chromosomes, because there's more than one chromosome now and you have to keep track of them. Again, we'll work our way up to doing that. But for some organisms, making another cell is their version of reproduction, and that's the important part here. Now, for multicellular organisms, we are concerned more with growth and repair, right? Any kind of situation where you need another cell, whether that's to get bigger or to replace one that's already been damaged. This is something we would go through mitosis. But before we go to mitosis, there's this thing called the cell cycle, because most of the time cells are in something called interphase. Then they will go into mitosis, but then they'll go back into interphase to live their lives, and then they'll divide again through mitosis and just alternate between those stages. Now, interphase has 3 steps, G1, S, and G2. And then mitosis has 4 steps. And then there's this other thing called cytokinesis, which is slightly different. And these are some vocabulary words that you might see. So we'll start with interphase and talk about controlling the cell cycle. G1 phase, as hopefully you remember, is for the growth phase. This is where the cell is just doing its everyday job, making proteins and doing the things that it is programmed to do by its DNA. But sometimes we might need new cells. And so there's this thing that we call the G1 checkpoint, a place where cells will not progress until they are given a chemical signal, right? Now, this is the little tricky part of it where we call cells that are not dividing in the G0 phase. This is kind of just a generic term for any cell that won't divide again. As you can see, muscle cells and neurons, but here's just a little image. If the analogy makes sense, that if you get the green light, you get to go past the checkpoint, but if you get the red light, or I would say that you would just never get the green light, then you would just be waiting for the green light over here in G0. Cells can sometimes go a really long time without going through the cell cycle. Imagine maybe like a bone cell, like a cell way in the middle of your bone. That one isn't going to need to make a copy of itself very often, but at one point maybe you'll get a fracture of your bone, and then suddenly it'll need to go in to make more cells. And so it would have been in G0 for a long time, but then suddenly it would go past G0 into the next phase. Now, this is where the cell communication part came in from the last video, that cells shouldn't do this unless they're told to do it, because you want to coordinate with the other cells in your body, right? And so we should only be growing when directed. This is a growth factor would be an example of a ligand, a signal molecule that is going to be released from somewhere. In this case, it's an example called PDGF, made by blood clots, right? So you get a cut on your skin. These little platelets are going to start to release this growth factor, and it's going to tell the cells around it to start to go to the next step, because it's like, hey, we need some new cells now. Again, to kind of remind that this is all about cell communication. So here we have our reception, and then we have our transduction going in here to a nuclear response, which in this case is going to produce a protein called a cyclin. And hopefully that rings a bell, that cyclins are pretty important. So an external chemical, an external signal, then was converted through the cell communication pathway into an internal chemical signal. Cyclins regulate the cell cycle. There's a bunch of them, and they're all associated with one particular step in the cell cycle. The basic idea is that as these molecules are created, it will eventually tell the cell, now go into the next step. So the cyclins I've represented like this, but there's also this part called the cyclin-dependent kinase, which I've represented like that. Now, the kinases are always present in the cell all the time. And these are the parts that are going to cycle, going to get made and then destroyed. And again, that's what this graph is showing, where the lines rise slowly and then they drop as the cyclins are being made and then destroyed. As they're getting made, the more and more there are, the more likely they will combine together with the cyclin-dependent kinase to make an active complex. This is actually the molecule that is going to go off and make the cell go from G1 into the S phase. But it needs the cyclins to be made to join with the kinases to make the active molecule, right? And then, so that's kind of like on this part of this curve right here. But then slowly after that, the cyclin will get degraded and the cyclin levels will go down and the activity that had been started will stop. Of course, at the same time, there's a new cyclin that's going to start to be made that's going to get us to the next step, because that one has a different job and tells the cell to do a different set of steps. So we get our signal, which results in cyclins being made that pushes us into the S phase, where hopefully you remember DNA synthesis happens. We copy our chromosomes, and then, because we need twice as many if we're going to cut them in half later. Then it keeps going into G2, where the cell is going to get ready for mitosis. S is about making more DNA. G2 is about making more organelles. I've got to mention really quickly that there's another organelle that kind of doesn't get talked about a lot because it's not for making proteins. They're called centrioles. They're these things right here, and they help to make fibers that are going to attach to the chromosomes and help them move around a little bit. So that's all something for mitosis that we'll get to later. Now, when you get to the end of the G2 checkpoint, we have, or the G2 phase, we have another checkpoint because we don't want to go to the next step unless everything is ready. Most importantly, our DNA. The DNA synthesis happened properly. If there was a mistake that was made during the S phase, and then we keep going into mitosis, we are now about to start making official copies of messed up, mutated cells. And so we want to avoid that as much as possible. So the cells shouldn't start mitosis unless they have met all the requirements and that they've gotten their CDK, active CDKs have been produced and tell the cell, okay, now it's time to go forward, right? So here's two more representations of the same idea as before, but they're more tailored towards this checkpoint. In particular, the CDK complex that does the work is called MPF, mitosis promoting factor is what that stands for. But the idea is that when you get these to be made, those will go and make the cell start mitosis and doing the phases of mitosis. You need these cyclins to slowly be produced over time. And once they finally get built up, then you can go to the next step, right? Then after you're done with that step, again, it's showing you that the cyclins get degraded, the CDK stays, but the cyclin is going to get recycled for parts, for amino acids and things to be used for whatever the cell needs, right? And so that would be, if the cell gets enough cyclins, it'll go into mitosis, the M phase. Now, I'm not going to talk about that one just yet because that leads to a whole other set of ideas. And I think it would be better right now to talk about what happens when you can't regulate the cell cycle. What happens when there's problems? What happens when there's problems is that you get cells that grow out of control. They start to grow when you don't want them to. They make a host of problems, which of course are not very good. And so but as it pertains to us, the idea is that cells shouldn't start to divide unless there are growth factors there, unless they were told specifically by some other mechanism, hey, it is time for you to make a copy of yourself. Cancer cells are going to do that, either because they think they're getting the external source signal, or they start to make the internal signals on accident. And so again, you shouldn't have to remember every specific detail, but a very common problem has to do with a protein called the RAS protein. Of course, that's made by something called the RAS gene. Here we have our cell communication pathway, where there's a growth factor that gets received by these proteins that makes then a conformational change, which then releases the RAS protein. As you can see, it's labeled as a G protein. You might have noticed that happens a lot. Those G proteins are pretty common. Now that's the one that triggers the cascade, which eventually says we want to make something. In this case, it's going to be a cyclin, right? But in a lot of cancers, a huge number of cancers statistically, this gene, the RAS gene, is activated permanently. It's gotten a mutation so that its shape is already in the shape to be active, right? It doesn't need that shape change to make it turn on. And so it starts to send the signals without this stuff happening, right? And so if this starts the signal cascade, the other molecules, the other proteins have no idea that this is a wrong message, and they just do what they're going to do. The cell makes lots of cyclins, and the more cyclins you have, then you start to go into the next steps of the cell cycle, right? And so here we are on our way towards having cells that are growing out of control. There's a lot of different ways for this to happen. They go into a category called proto-oncogenes. The onco part means cancer, and proto means like before, right? So like these are genes that you have that do a real job that you need, but if mistakes happen, if they get overexpressed usually, the cell cycle starts to go too fast, and they just start to copy and copy and copy. There's a wide variety of things that could happen. In this case, a promoter from, say, a more active gene gets accidentally moved. These things can jump, they get cut out and then it's inserted in certain places. And suddenly a gene that was only supposed to make one starts to make 3 of that protein, 3 times as much as you need. And that should maybe cause the cell to divide 3 times as fast. I'm sure that's a generalization, but that's the idea. Along the same idea, instead of having a new promoter, you just get a straight up duplication where you have 3 sets of the same gene to make 3 times as much stuff. You could get a control element. We didn't talk about this yet in the review, but maybe you remember that we need these transcription factors to combine with those control elements. And if all 3 get bound to their proper molecule, this will activate the gene. But then maybe this one that's showing right here is already mutated, so you only need 2 of those molecules, and so you get accidental activation and you get extra stuff being made. The RAS gene, the example of the RAS gene is more like this one, where a change in the protein, within the protein, makes the protein change its shape. And so that change happens to be the shape that results in an active protein, right? Instead of being a normally inactive protein, it would be starting as an active protein thanks to the shape change. All of these things will make too many chemicals, cyclins, which will then lead to too much division, which leads to all the problems with cancer. But you're supposed to be able to stop this. And so there's other genes called tumor suppressor genes. They are slowing down or inhibiting the cell cycle. Again, you shouldn't have to memorize any, but P53 is the most common one, and it is very famous. And so P53 is the example that gets used the most. 50% of cancers, as you can see, that's a lot of different types of things that could go wrong. But in 50% of them, this is something that has gone wrong. So that's a pretty important gene. Now, its job is to halt cell division if it detects damaged DNA. How it does that is way beyond our pay grade. But the idea is that if it detects damage, it will start a process that breaks down the cyclins, right? So we are down here at, oops, I thought there was going to be a thing, right? Down here. And so you need cyclins to get past the checkpoint, but the P53 stops that from happening, you don't get any cyclins, then you won't go past. It also has a job of starting something called apoptosis, where the cell will die off. It'll kill itself, or it'll ask the immune system to kill it for the greater good, right? And so that's kind of what's being shown here, that if you get DNA damage, you should either repair it and then continue on, or if you can't repair it, you should apoptosis, you should die. And that way the mistakes won't carry on. But if we don't have the protein here, the mistake doesn't get caught. It goes to the next, it gets completely copied, but then it makes a copy of itself. And so now, of course, you have two cells that are mutated, and then this will keep happening. As that happens, more and more mutations can happen. And so maybe this one is on its way to being a true cancer cell that has growing out of control and has a lot of these other problems that cancer cells have. That again are kind of going to be beyond the scope of what we are going to do in this section of the unit, right? And so kind of just to wrap this part up by talking about what causes cancer, because this will, again, just hit home the idea that we're talking about genes, which make proteins that do their thing, right? And so this group, radiation and chemicals, these are things that are going to just break your DNA. They're going to hit the atoms and make them fall out of the molecules. They're going to make reactions happen where the molecules split or something, whatever it's going to do. But these are going to attack your DNA. We already know that heat destroys biological molecules, so that's bad. Age, again, I wanted to point this one out, even though we haven't done the review, was that mistakes happen. They happen a lot. You can't stop the mistakes from happening. So every time a cell copies itself during mitosis, you have the possibility of making a bad mutation, which leads to a proto-oncogene being mutated, and then you start on your way to getting cancer. So the idea would be that the older you get, the longer you live, the more chances that one of those mistakes is finally going to happen to you. Sometimes you can be born with mutations. Sometimes if those mutations that happen naturally over time happen to occur in one of your sperm or egg cells, you could lead to your child being born with a RAS gene that is already mutated, right? And so this is already going to set you on your path to having cancer. Now, the thing about these two is that the proto-oncogenes are important, but the tumor suppressor genes are even more important, really. Animals, they found that animals like elephants that very, very rarely get cancer, they don't have any special ability that our cells don't have, other than they have a lot of copies of the P53 tumor suppressor gene. So these mistakes will happen all the time, but hopefully you can catch them with the P53 and stop them from snowballing into a bigger and bigger and bigger problem, right? Because in the end, this is all just cell signaling and controlling the steps of what's going to happen. All right, so as it has been approximately 17 minutes, that's probably enough of listening to me for right now. I will make another video soon about the steps of mitosis in particular. So until then, let me know if you have any questions.